Hi everyone, welcome to our webinar on Roseate Spoonbills. We are just waiting for everyone to come into the presentation and then we will go ahead and get started. Thanks everyone for joining us. We're just waiting for everybody to come in via the registration link and then we will go ahead and get started shortly. It's 4 o'clock, but we're going to give everyone just an extra minute to come in. So just sit tight and we will be starting pretty shortly. All right, people are still trickling in, but we're gonna go ahead and get started. Thank you so much for joining us for Roseate Spoonbills in Florida Bay, a pink canary in a coal mine, presented by Dr. Jerry Lorenz, Research Director for Audubon, Florida. My name is Erica Zambello, and I'm the Communications Director for Audubon, Florida. And I just wanted to do a few housekeeping things before we get started. Everyone is muted and everyone's video is off. However, we will have the chat box running. So if you have any questions that you're worried about forgetting or you want to just ask it during the presentation, go ahead and put it in the chat box and I will be monitoring it and we will get to your questions at the end of the presentation. For all the folks on Facebook Live, hi, welcome. We are also answering your questions as well. So just put those in the comments and we will do the best we can to get to your questions. This presentation is being recorded, and so if you miss something or you don't want to take notes, don't worry, we're recording it. We will upload it and send you a link to the recording. Jerry does have an arrow cursor within the presentation. Right now it's just about by the bill of the Roseate Spoonbill, so he will be using that to point some things out. And I believe that's it for me. So without further ado, Dr. Lorenz, thank you so much for being with us today.
Well, thank you, Erica. Yeah, I'm coming to you from the Keys, and where I've been studying spoonbills and spoonbill ecology for the last 30 plus years. And one of the things I like to start off with is I'm not an ornithologist. I'm a marine ecologist, but primarily a fish guy. It's what fish is what got me into studying ecology. Now, it was strange that I got a phone call, a job interview from the Audubon Society, and I was kind of like, wow, maybe they're coming around to my way of thinking. And so I started interviewing for the job, and what was brought up to me was, okay, we're doing this fish study in South Florida, and it has to do with spoonbills. And I was going, now wait a second, the only spoonbill I know is this. This is a paddlefish. It's also known as a spoonbill catfish. And it is found only in the Mississippi basin. And so I was thinking to myself, well, what could I possibly be going on in South Florida with this fish? Well, I thought it is a fish that's aquacultured. Maybe they're using it in aquaculture. So I ended up getting prepared for my interview by reading this. Needless to say, I was quite surprised when I got here and this is what we were talking about. So I had to adapt on the fly. I still managed to get the job even in my ignorance. And this was all new to me. But, you know, I just thought, well, okay, you know, I come down and all of a sudden I'm just knocked off my stool because my job is to identify dead fish in spoonbill vomit. And that's where I started. But it has led to quite the interesting career. So where this starts is with the Everglades. Now on the left over here is a satellite image of southern Florida taken in 1492. And it just shows the river of grass, the continuous flow of water from Lake Okeechobee southward into the Gulf of Mexico and into Florida Bay. And through drainage and management practices on the right, we've completely compartmentalized the entire Everglades. And so these are management units that you see. And these management units are kind of managed independently. And so there's no strategy for getting the water to do what it's supposed to do. And that's ultimately to get down here to Florida Bay. And of course, this has caused some very dramatic ecological changes in Florida Bay. Now, this is a blow up of the main freshwater flowway into Florida Bay. If you see my cursor, this is Taylor Slough, and this brings fresh water to the Florida Bay estuary. An estuary is where freshwater and saltwater mix, and it makes them highly productive, biologically speaking. And so this northeastern part of Florida Bay is largely where I focus my work. And it has changed quite a bit. This slide, the green lines just represent the area of mangrove dominance. A mangrove is a salt-tolerant tree. So this is kind of an indication of how far inland the marine system penetrates. In 1948, it was right on the edge of Florida Bay. And then slowly, by 1967, it creeped a little ways inland. 10 years later, it was way inland. And 15 years after that, it was 3 to 4 kilometers inland. And this cannot be explained by sea level rise, even though sea level was continuously rising during that period, not at that rate. And so the only explanation for this is that freshwater flow through Taylor Slough was cut off. And it was, in fact. And when I arrived in 1989, this massive seagrass die-off was ongoing. It began in 1987. On the left is a picture of what a healthy seagrass bed in Florida Bay should look like. It made the bay just look emerald green on the surface. It was spectacular. But over the course of several years, what you see on the right, all these cross-hatched areas were thousands of acres of where the bottom was just denuded with seagrass. It just died. And there's a scramble to figure out why. But of course, all that seagrass decays as well. And what you end up with are these algal blooms. And these blooms, they persisted. This was in 2007. So this was 15 years later. And yet they were still persisting. And as you can see here, I like to say Florida Bay is not Las Vegas. What happens in Florida Bay doesn't stay in Florida Bay. These are the mainline keys here. So this is Key Largo, Plantation Key. This is where down by Marathon. And you can see it went out in between the keys, out to where the reef tract is. And when you look at this, this is a picture of Tennessee Reef, offshore about 3 or 4 miles. And this just, from this area, it just looks like the reef. But when you blow it up close, there's this green slime that's oozing out of Florida Bay and covering our reef. And of course, this is a real problem for a tourist-based economy. People come here to fish and to dive and to snorkel. And if it's unpleasant in the water, it doesn't help our economy much. So after doing this for many years and looking at the scientific literature, I published in 2014 just a review paper. I looked at all the publications that dealt with the vertebrates that live in Florida Bay and how their habitats changed due to this lack of freshwater flow and increased salinity. And you're not expected to read this table. This is just a table from that publication. And in the first column, it lists all of these species or groups of species that were negatively affected by the changes in water flow. And just to go through some of these, the American crocodile almost went extinct. Mangrove terrapins went into a dramatic decline. We couldn't hardly find any of them. So there's 2 reptiles, 2 mammals, bottlenose dolphin and West Indian manatee. Florida Bay was the largest place for them to aggregate during the dry season. When there wasn't a whole lot of rainfall, they would come in here for fresh water. And although they still use it, the numbers have declined dramatically over the last 50 years. And then there are 5 species of bird. 4 of them I present here: the great white heron, osprey, brown pelican, and bald eagle, all crashed in Florida Bay over the course of about 20, 25 years. And of course, the 5th one is the spoonbill. Now, the other things that we documented, and this was kind of my research as well, is that the prey-based fish populations that all those other animals ultimately feed on through the food web also went into decline. The numbers just weren't what they were historically. And of course, without the prey, you also don't have the game fish. And this is, again, an economic driver. And what I pictured here is a healthy seagrass bed. You take that away, in these 2 species, the upper one being the common snook and the lower one being the redfish, they wanted a tailspin too. Now, a snook is one of my favorite fish. And when I took this job, this is the fish that I really wanted to come here and study. The problem is that you can't follow snook around. You can't get really good numbers. You can't see how their offspring are doing. You can't see, you just can't do a nice ecological study on their numbers going up or down. It takes, over the course of years. But there is a nice surrogate. So this is a guy who just caught a snook. And here in the background is one of my little pink buddies. And so he caught this fish in the same water that this bird is feeding. And snook tend to regurgitate what they've just eaten when you hook and line them. On the right of the screen here is the line going in with the hook in it. And you can see kind of in the dark cave of his mouth, there's a whole bunch of little fish that the snook has been feeding on. So when you take those fish out, what you see is encircled in red. That's the primary species that spoonbills feed on. And spoonbills do feed on fish primarily, and especially in Florida Bay. The ones in the yellow are prey items for a little bit bigger birds, such as birds that take a little bit bigger food, such as like great egrets and such, the herons. So I just started looking at the spoonbill, even though I was hired to study their diet. I just kind of had a change of plan. And you call this a roseate spoonbill. I think it's a pink snook. So that's how I kind of took over the spoonbill program. And that happened in about 1993, 94. So I took a different course, and it led me on some interesting paths. Now, spoonbills feed by tactile location, so they feel for their food. This is a picture of a foraging spoonbill. And what they do is, instead of like a heron that visually sights its prey and then strikes, spoonbills put their bill in the water, walk forward, they look rather drunken, and they wave their bill back and forth. And when they encounter a prey item, the bill snaps shut, they swallow it, and they move on. And so I kind of think of them as little vacuum cleaners. They're extremely efficient because they don't have to sight their food. They're just walking through the water, and they'll hit fish after fish after fish. Or they'll eat shrimp, and they'll eat crustaceans, and they'll eat mollusks, things like snails and such. But they are primarily designed to eat fish. And in Florida Bay, looking at the prey studies that the birds eat, these 2 small fish make up the vast majority of their diet. In the 1990s, we did an analysis on spoonbill diets, and these 2 fish made up about 70% of their diet. And you can see they're only 5 centimeters long, or about 2.5 inches, or about 2 inches. And so these are relatively small fish, and that's what spoonbills feed on. Now, in the Everglades, what we have is a situation where we have a wet season, dry season. And the wet season goes from June to November. And during the wet season, the entire Everglades is flooded, the entire landscape. And so these little fish spread out all across the landscape into what's essentially a low predator environment. And so there isn't a whole lot of predation pressure on them. They have very quick generation times. So for example, one of this fish, a mosquito fish, from the time it's born to the time it reproduces is only 6 weeks. And so they have multiple generations over the course of a wet season, and the numbers of these little fish grow exponentially. And so at the end of the wet season, you just have a tremendous amount of these little fish spread across the landscape. But then the dry season starts in December, and water levels start to drop. And these fish don't want to get stuck out on those wetlands that are going to go dry. So in our studies, We demonstrated that where spoonbills feed, this occurs right about 13 centimeters, about 5 inches. So when the water gets to about 5 inches on that wetland surface, they head for deeper water, whether that be ponds or alligator holes, or in the case of spoonbills, tidal creeks or coastal creeks. And as it goes on, they become highly concentrated. And this is when the game fish and the wading birds exploit the resource, is when they are this available. And the wading birds themselves gear nesting to this because they're going to need a reliable source of food to feed their young. Now, this isn't a great picture. I took this picture in 1990. It's just a picture of my hand in the water, and about 10 feet away from me, spoonbills are foraging. And so I put my hand in the water, and all that you see there, those are just little fish. And if I lift my hand out of the water, I can collect them. Now, think about that. The spoonbill is evolutionary. It has evolutionary designs to capture these fish. I do not. If I can capture those fish that quickly, you know what spoonbills are doing. And they need that concentration because spoonbills lay 3 eggs, and the eggs are about the size of a chicken egg. So when the chicks hatch, which this chick just did, they're about the size of a chicken egg. And they grow very quickly. Within 6 to 8 weeks, they are 2/3 the weight or maybe more than the adults. And so the adults, having to feed 3 chicks from the size of a chicken egg to this size, have to be able to go and get food, get it now, get it quick. And it's almost a conveyor belt. Go get food, come back, feed the young, go out again. And that goes on until the birds fledge from their nesting colony. Now, as we all know, is that most birds in Florida and in most places were hunted during the plume hunting era. And the end result was that spoonbills were extirpated in the United States. And while all the other birds, once the laws were put in place ending this practice, all the other birds came back. So by the 1930s, we were having nesting colonies in the Everglades of herons and wood storks and such in the hundreds of thousands, but there weren't any spoonbills. That changed in 1935 when Audubon wardens discovered 5 nesting pairs of spoonbills on Bottle Key in Florida Bay. John Baker, the president of the National Audubon Society at the time, wanted to know why certain species were not covering after the hunting practices were removed. And so he tapped this gentleman. This is Robert Porter Allen, Bob Allen. And he told Bob he wanted to know why spoonbills don't recover when all the other wading birds have. So he sent Bob to Tavernier. And okay, so Bob moves from New York City to the backwater of Tavernier in the Florida Keys in 1939 to study spoonbills. And although Bob did work on whooping cranes and flamingos as well, he headquartered himself for the rest of his life in Tavernier. Now, Bob died in 1963. He was the first science director, and he established the Tavernier Science Center, which about 15 years ago was renamed the Everglades Science Center, that I'm the director of now. Bob is fascinating. He wrote 2 popular books. I highly recommend, if you're interested, you read about him. There's also an account of him saving the whooping crane that is also available. Now, when Bob was doing his work, this is where the birds nested in Florida Bay. The red dots represent the nesting colonies, and the blue circles represent the little more than average flight distance of a spoonbill from its nest to go get food. This is about as far as they're going to fly. And in this particular picture, what you see is all of these colonies overlap right here, and I hope you can see that, but it's at where Key Largo ends and Plantation Key begins. And that's where my office is based. And what happened here is that the colony numbers went up. Now, Everglades National Park was established in 1948, and Florida Bay is part of the national park. And with that protection, we see this rapid increase in the number of spoonbills over the course of a decade. And that's the white line. Now, the red line is a single colony, the largest single colony at the time, and it's called Cowpens Key. And you can see it rapidly increased to having all, in some years, all the nests in that region of the bay. Now, this is just a map of the area, and this is Tavernier Creek, and this is Key Largo, this is Plantation Key, and here's Cowpens Key. Tavernier is here, and that's where Bob lived. And so from beginning of about 1958, during nesting season, Bob would take his boat out, come out to Cowpens Key, and observe the birds. And one of the things he did was quantify where the birds were flying to and from the colony. So he'd just sit there with his binoculars, he'd count the birds, note the compass heading that they were going to, and he did this for thousands of flights. Then he would sleep out there and do the same thing in the morning. And he did that pretty much every day for 5 years. Unfortunately, shortly after collecting the data after the 5th year, he suddenly died. And so his notebooks were kind of inherited by the office. And in the 1990s, I was just going through them because I'm fascinated by this man. And I'm looking at this data going, wow, that data is really, really interesting. And there's a note from Bob in there that says, I wonder what's going to happen to these birds with all the development on Plantation Key. I wonder if this colony will continue to exist in a few years. Well, that was rather prophetic. So I took that data and I made these pie charts, and this takes a little bit of explanation. So the middle of the pie chart represents Cowpens Key. The color intensity represents the percentage of flights. And again, these are thousands of flights in the direction they're heading in. So for example, in 1957-58, about 70 to 80% of the flights were going to the east and the southeast. Highly directed flight lines. These birds were going to a certain place. And where they were going, and now this is a slide, this is an aerial photograph from 1945 on the left and from 1964 on the right. And I had to lean these slides, the pictures over to get them on this slide, but north is diagonal. And so from Cowpens Key to the east is this very big wetland, and then to the southeast is this very big wetland. Well, this very big wetland was turned into this canal front subdivision in 1958. By the way, my office is in that subdivision. And so what happened was in 1959, the birds quit flying directed to the southeast, and almost all the flights were going to the east, again, directed that way. So now we could come back again. Here's this wetland. It was converted to this development in 1959. And by 1960, the birds were flying in a random pattern. And the numbers of nesting birds started to go down. 61, 62, 63, they're flying every which way. And by 1963, the colony had collapsed. As you can see from this slide, this is where Bob collected the last data. And when we started collecting the data again in 68, they were gone. So Bob also collected information about how well they were actually producing, how the chicks were doing. So this is an average nest production. It's just average chicks per nest. And prior to 1971, the average chicks per nest for all of the colonies was 2 1/4 chicks. So out of every 4 nests, we got 9 chicks fledged. After that, it dropped to 0.25. So 4 nests produce 1 chick. And that is not a sustainable population. That's a population that's going into severe decline. So what we learned from this is that alterations to the foraging grounds is what's important. Is if you take the groceries away from the birds, they have no choice but to move someplace else. They can't raise their young. And so they move. And where they move to is here in the northeastern part of the bay. They move to these colonies that are represented by red dots. And again, the rings around it are the flight distance. And so these birds were pretty much foraging in this Taylor Slough and adjacent wetlands habitat. A study that was done by my predecessors, they captured adult spoonbills on nests and outfitted them with radio tags, and then they'd go up in an airplane and find the birds. And where they were finding them foraging was all along this coast. And this is from 2 different major colonies in northeastern Florida Bay. And so this just demonstrates that this is where these birds, this is the wetland that these birds needed to forage. And we know what happened here because the percentage of nests in the Southeast Bay, it crashed. The numbers went up in the Northeast Bay. So the birds just moved a little bit north. And now looking at just the Northeastern, I'm going to divide this into 3 periods. This first period is when most of the birds were nesting in the Southeastern Bay, but we're still getting 40% from somewhere between 10 and 40% of all the birds in Florida Bay nesting in this area. Now, during this period, we really hadn't influenced Florida Bay that much as far as canals go. So it was kind of a natural system, even though the Everglades was already being drained, the southern part was not. And so the way water flows to Florida Bay is this is the main flowway, Shark River Slough. And there's a ridge on the west side. And then there's this low-lying upland called the Rocky Glades on the east side. And the water during the wet season would kind of bottleneck here where it got pinched. And water levels in this area would get high enough to cross the Rocky Glades, flood it, stream across it, enter what we call the headwaters basin, and then ultimately flow through Taylor Slough into Florida Bay. And that's the way that the system worked historically. So then along comes the second period where the birds had to move to the northeast. Now, right about the time this happened, this canal system was completed. And what was happening here between 1960 and 1965 is the orange in this picture is agricultural lands. And there was always seasonal agricultural here going back to the early 1900s. But this canal system allowed for year-round farming. And so it was operated to that end. There wasn't much concern for the Everglades National Park, or Florida Bay. And so that's when the damage started. And then finally, this third period where the number of spoonbills in northeastern Florida Bay just plummets. And during this period, what had happened is this system of pumps and canals increased in size. Is called the South Dade Conveyance System. It was built between 1962 and 64. It increased fourfold the size of the canals that were drained in this area. And it also included these massive pumps. And just to show you the difference, this is a picture of one of those pumps. Now, this canal in the background is what the canal system was prior to 1962. This is how big it was made after 1964. And this pump is the S331 pump, can pump 1,250 cubic feet per second of water. So that's a swimming pool per second or more. And the other thing in this photograph that you can see is this used to be Everglades. It is now farmland. So they literally dried the Everglades. And this altered both the quantity of water coming to Florida Bay and where it arrived at Florida Bay. So it reduced the amount by 3/4, but it also moved the water away. The green arrows indicate where it's supposed to go, flow through Taylor Slough, end up in this northeastern basin, and have a long residence time. So it keeps the salt water, the salinity, low. Well, the canal system routed it around to right along US 1 going into the Florida Keys, and it wrapped around Florida Bay, and it was still only 1/4 of the water. And so our estuary was cut off from its freshwater flow, essentially. Now, this is just a graphic that, again, shows those 3 periods. The first period is where we had natural flow. And this is the wet season average maximum water level in Taylor Slough and the dry season minimum. So we have 1.5 or 1.25 meters of water in the wet season, dropping to 1/4 of a meter in the dry season. This means lots of fish. You got a lot of water in the Everglades. That means the wet season, there's a lot of fish. This means it's dry. That means those fish are concentrated. This is a highly productive system. In the second period, the wet season got lowered by 1/2 a meter, but it still dried out. So we didn't have as many fish to start with, but we were still concentrating them. In this third period, you flatline the whole thing. You don't have enough water to make fish, and the water's too high to concentrate what is there. And this had a profound effect, again, on production, the average number of chicks per nest that were produced. In the first period, that was 2. So 2 chicks per every nest. In the interim period, it was 1.6 or 1.4. So that's still pretty good. That's a growing population. After the pumps came in, it was 0.6. You got to produce at least 1 chick per nest to be considered a successful nesting year. And so this is a failing population. And if you just look at the percent of years, again, 100% in the first period, it's about 75% in the second, and only 4 out of 10 years in the third period. So the spoonbills are telling us there's something wrong out here. Now, this is the actual number of spoonbills in northeastern Florida Bay in red, and the total number in Florida Bay in blue. And you can see that after those pumps were put online, northeastern Florida Bay plummets almost down to 0 in 2011. There was this jump in the total number because the birds moved to the western side of the bay, but that had its own problems. So there was kind of a delay in the collapse of the rest of the bay. But now it's kind of jumped back up and stabilized. And I want to talk about that in a few minutes. So my job was to get to the bottom of why these changes in water flow were really altering these fish. We didn't know all this stuff that I just explained to you. So in 1990, I started looking at the prey base of the spoonbill, and I invented this. This is a 9-meter square drop trap that I use in mangrove habitats where spoonbills feed. And when I pull the trigger on it, by the way, this is me when I was young, I actually had hair. It falls in the water and surrounds a 9-meter square area, sometimes including a small mangrove tree. And this creek type habitat is where the spoonbills feed. And then I would clear the trap with a toxicant called rotenone, which is literally sawdust from trees of the genus Daris. It shuts off the gill function of the fish, so they come to the surface and I can capture them. But it biodegrades within about an hour and a half at the temperatures that we have. And this has a blue dye in it that I put in just to demonstrate that I have a flow curtain on my traps. And out here, you can see there's no blue. So I contain this for the hour or two it needs to degrade, but it works on the fish very quickly and I can collect them when they swim to the surface. So I get a nice quantitative measure of how many fish there are under what conditions. In using all this information, I developed what's called a conceptual ecological model, or a CEM. And this is that model. Now, it's simply a flow diagram. Is that water management practices and rainfall determine the salt content, the salinity in that habitat, and it also determines the water levels. The salinity, in turn, determines the submerged aquatic vegetation, the plants that grow on the bottom in this water and provide habitat for fish. Salinity also directly, physiologically affects the prey-based fish. Some fish are more tolerant to salt than others. Water levels, as I showed you, determine how abundant the prey are, and then it determines how much they concentrate. And it's this concentration that determines how successful the spoonbill nesting is, and that determines the number of roseate spoonbills. So just to go over some of these steps, is one of the studies that we did showed that when you have a freshwater community of fishes that's in the blue, you have a lot more fish and a lot bigger biomass of fish than you do even with a transitional, just getting a little salt. And then as you increase salinity, it continues to go down. So the fresher the habitat, the more prey you're going to have. Now I've already showed you this determination that the fish concentrate at about 13 centimeters. So this is an aerial photograph of one of the sites we work at. This is our Joe Bay site. And those are my nets. The squares you see, those are my nets set up and deployed. There I am collecting fish. But you can see these are what we call the flats. They're the ephemeral wetlands that are wet during the wet season and dry out during the dry season, forcing the fish into this central creek. Now, we identified all the sites we looked at because we were doing aerial surveys, and we would see things like this. This is a picture from 1990. And these are all roseate spoonbills feeding at where I sample fish at the Highway Creek site. So this is how we determined where we were going to go collect fish. Where were the birds feeding? Go there. And what we discovered, this is something, again, that is actually from my dissertation work, but I divided the years that spoonbills were successful, where they produced more than one chick per nest on average, from years where they didn't. And this is water level at the 4 sites I was working at the time. And you notice that when they were successful, the water level at all 4 of these locations was below 13 centimeters. And the fish abundance was really high, both this is density and this is biomass. When they failed, the water was invariably above that 13 centimeter mark, and there were no fish to be had. And so they failed. So the other thing that this did is it not only altered the distribution of timing, but it altered the timing. And so here's your farmland up here. If it rains at all during the dry season, they want that water gone quickly. They don't want it. You know, we get some pretty heavy rainfalls even during the dry season. And so they shunt this water south. And so it immediately ends up in this wetland where the spoonbills are feeding. So it can change the water depth very quickly, which is depicted here. This is the 1992-93 spoonbill nesting season. This is the date when spoonbills would have laid eggs and begun hatching. And this is how long it would take to raise their young. And this line is at that 30 centimeter mark. And this is just the daily water levels from 2 of our stations. And so the spoonbills were great at predicting when the chicks should hatch. The water level just got to the point where the prey fish are concentrated, we can feed our young. Well, then those pumps turned on, we got some rain, and it went above, and we got more rain, and we got more rain. The dry season of 1992-93 was a good rainfall year. And so the bottom result was the entire spoonbill nesting population failed. I don't believe we found a single chick in northeastern Florida Bay in 1992-93. Complete failure. Now this, unfortunately, is a video, and during our warm-up, we couldn't get it to play, but it is just a picture. It's a short 8-second video that I collected. It's heartbreaking. What happens is the adult spoonbills, they fly off to go find food. And if they can't collect enough food to feed their young, they just keep hunting. And then the second adult leaves the nest. And eventually these spoonbills chicks become so weak that they fall out of the nest. And invariably spoonbills nest over water. And so when I found these 2 chicks, they were struggling in the water, and there were 150 nests at this particular colony. I believe this was in 1994. And that means 450 chicks. And if I turn this camera around, there was literally a rack line of dead spoonbill chicks all along the shoreline. I think we counted 10 chicks that survived this. And those 2 that I have pictures of, I include in that number. I'm not the cold-hearted bastard my employees would tell you I am. I rescued those 2 birds and took them to the local rehab center, and they were released as subadults a couple months later. So I used all this information to, again, publish, and this was in Ecological Indicators. And it's a journal that looks at how can you look at certain things to tell whether an ecosystem is doing well or not. And spoonbills were one of 13 species or groups of species that were chosen to report to Congress, the US Congress, about how Everglades restoration was going. And so every other year we report on these indicator species. And SERP stands for the Comprehensive Everglades Restoration Plan, funded by the US Congress and the state of Florida. And so just to give you an example of how we do these reports, the congressmen and senators aren't going to sit down and read a 20-page report. So it's got to be very quick. And so this is an example of something that my colleagues at Audubon and I at Audubon, Florida, put together this year to convey this message very quickly. And I'm not going to go through this whole thing, but just to give you an example, there's a water level metric. And so here's that water level metric. And it just shows this line is at 13 centimeters. The gray in the background is what's normal for the last 30 years. So this is about where you want to be. You can see last year in the lines at the top are the maximum and the minimum. So last year we set a lot of maximum water levels. Water levels were extraordinarily high. And it only got below that prey concentration threshold for about 9 days. So that's really bad for spoonbills. So it gets a red light. This is, a senator can look at this and just go, okay, red light, it's bad. Or he can look at it and get the details. Same with salinity. Now salinity is yellow because it's a mixed bag. The normal says that we have freshwater conditions usually be starting in July and lasting through about February. Well, last year we had these peaks, and again, sometimes setting the maximum for a particular date. And those peaks are really bad. The spikes in salinity are what really cause problems. So that's bad. But look how far into the wet season we had freshwater conditions, to April. And that's unusual. And so there were good things and bad things about this. So we scored that a yellow. So we also, in the mid 2000s, we started a banding project. We wanted to see where these birds were going, see how they move around the state maybe, or just see how they move around Florida Bay. We instituted this banding program in both Tampa Bay and Florida Bay, where we had large populations of spoonbills. And we banded about 3,000 chicks. We've now started banding in St. Augustine as well at a lower level. But to just give you an example of, you can look it up on the Audubon website. You can just Google report a banded spoonbill or tag spoonbill, and it'll come to the website and you can report it. And this is an example of the kind of report I get. Now we just blacked out the names and the email address and the phone number, but this is a legitimate report. This person found a bird with a brown band on the left leg. It was horizontal orientation. The code was WR. It was seen on this date at this time in Hermsburg County, and the location. And that's a really nice thing to get in the mail, in your email. I love seeing these. But the problem is mostly what I get is spam. So this is just being spammed, and it's, by generic Viagra. And you can quickly tell that, the phone numbers are made up, the emails are made up. They don't, date of sighting is never there. But then I got this one. And this one is crazy because there's a real name. There's a real email address. There's a real phone number. It says a tag, and it gives me an alphanumeric code. Those other ones never do that. But the alphanumeric code is only supposed to be 2 to 3 letters. And then it says Lake of the Ozarks, mile marker 87. And then there's this. The spoonbill tasted delicious. Now, I was infuriated. So I didn't want to talk to this guy. So I had a graduate student that I was working with call him. She's very nice. I can not be very nice. And it turns out it was this. They have a banding program for paddlefish in the Mississippi Basin. And so I just found it humorous that it comes back around. It's like a boomerang. Okay, so we also satellite tagged 20 adults from 2006 to 2010. And I'm not going to give you a whole lot of graphs. I'm just going to tell you what we found out with the banding and tagging program. Is that they live to be about 25 years old, we think. That's based on a recapturing of an 18-year-old. It was actually 16 when we captured it. But we put a tracker on it. And at 16, we just coincidentally caught an already banded spoonbill, so we knew its age. And we tracked it for another 2 years. And it probably didn't die, just our transmitter died. The nesting in Florida Bay, we calculated it at 0.6 chicks per nest in less than 4 out of 10 years. But the banding resites suggest it's even lower. So even though the chicks were fledging, they weren't surviving. And one of the most important things is they exhibit a high degree of nesting fidelity to their natal colonies. This just means they come back to where they were hatched as adults to breed. So salmon returning to the same stream, turtles returning to the same beach, spoonbills return to the same bay. So birds from Tampa Bay went to Tampa Bay. Birds in Florida Bay came back to Florida Bay. And the implications of the banding program are that in Florida Bay, the birds hatched prior to when the damage was done in 1984 are dead. And low nesting success means that there's not many adults. So this explains this decline in the number of spoonbills in Florida Bay over this period of time. Now let's look at Tampa Bay and Florida Bay and compare them. Nesting began in Tampa Bay in about 1973. There was only one or two nests. In 1976, 77 is when they started to increase. And so the trend is upward. And by 2005, 2006, there were more spoonbills in Tampa Bay than in Florida Bay. Florida Bay in red, Tampa Bay in yellow. So let's take a look. This is Tampa Bay. This is the Richard T. Alafaya Bank Reserve. This is where the largest colony of spoonbills in Florida is, or was. I'm not really sure right now. It's getting close. But there are other colonies spread around the bay. But look at this. This is MacDill Air Force Base. This is a Cargill plant. This is urban sprawl. You don't see many wetlands in this satellite image. This is Florida Bay. This is Turnkey. This, at one point in time in the late 70s, had 600 nesting pairs on it. This is Everglades National Park. You don't see many houses. You don't see many Air Force bases. This is a national park. Spoonbills are doing better in an urban environment than they are in a national park. And that's a good thing. But the bad thing is it tells us our national park is really sick. So it has an economic factor as well. The Florida Keys, this is what we sell. I said this earlier. We sell our coral reefs. We sell Florida Bay. We sell fishing. We sell our wildlife. That's what people come here for. And so the economic impact of restoring the Everglades was estimated by the Everglades Foundation to be a 4-to-1 return on investment. I'll take that investment every time. You increase jobs, you get higher real estate, you increase people coming here for fishing, wildlife hunting. So Everglades restoration is a good deal. And just some dollar figures. Estimated in $2,006, the annual user value of water-related activities just in the Florida Keys, just in Monroe County, was $660 million a year. And Florida Bay is the nursery ground for a $59 million a year shrimp fishery, a $22 million a year stone crab fishery, and a $40 million a year Caribbean spiny lobster fishery. So without Florida Bay, you can just take those fisheries right away. So this is not just about the pretty pink birds. It's about our economy. So what are we doing about it? Well, Everglades restoration. This is a simple diagram. This is what the Everglades was. This is what it is. And this is what we're trying to turn it back into. We're trying to restore flow going to the Gulf into Florida Bay. So how's that going? Well, this is the, again, nest production of spoonbills from 1982 to 2005. That's a 23-year period. The X's indicate years where we don't have any data. So now it's 20 years. Out of that 20 years, the red indicates where they did not produce one chick per nest or failed. So there are only 7 years where they were successful. That means 2/3 of the years that when I started working on this, I watched those spoonbills die. And that was painful. But now, let's see what happened since. Since 2005, we've only had 3 failed years. Spoonbills have been very productive. So what happened? Well, the Comprehensive Everglades Restoration Plan becomes law in the year 2000, approved by Congress. It took about 5 years for that plan to kick in. And where it really kicked in was this thing called Recover, which is a monitoring plan. So up to that point, water managers only looked at flood control and water supply. This mandated that you also had to look at the Everglades and at the environment and what your management practices were doing. And so they started listening to the people who were doing the work, like myself. I remember having weekly conversations with one of the water managers. And I just say, if you turn on that pump, you're going to kill my birds. Just that simple change resulted in a big deal. But then projects started coming online. And this is the C-111 Spreader Canal project. And what this project was designed to do was push that water back away from the canal going down US 1 and push it back into Taylor Slough. And preliminary results show it's pretty effective at that. It doesn't work in drought years so well, but in a normal rainfall year, it works very well. And so you can see, again, the population kind of rebounded and stabilized. It's not as high as it was, but 200 to 400. Well, there's a reason why it's not shooting back up at this point, and that's because of sea level rise. This is the over 100-year record of water levels, mean low water, in Key West Harbor since 1913. And steadily this water has been going up. But what's most alarming is the last 20 years. It's going up very, very quickly. And this has an impact on, it pushes water into Florida Bay too. And so it's changed the habitat where these birds feed. So this is just, again, water levels from one site. The mean from 2000 to 2010 is in black. And the pink line, again, is spoonbill nesting period at 13 centimeters. And you can see that prior to 2010, much of the year, especially during nesting season, water levels were below the prairie concentration threshold. 2010 to 15, it kind of started bouncing back and forth. By 2016, it was barely touching it. And if you just look at the number of days out of the year that prairie were concentrated, where water levels were less than 13 centimeters, 1990 and 91 is the first year I was working here. Well, the first year I was actually collecting data. It was a drought year, and 200 to 250 days a year, it was below that. That's not good either. But then when the drought ended, you ended up somewhere between, say, 50 and 150 days a year where it was below that concentration value. And then along comes 2012, 2013, 2014, 2016, there were 15 days. Now I told you there were 9 last year. And so this has, the birds they're recovering, they're doing very well, but they're not doing it the way they used to. Things have changed. And what has happened? Well, these are the 2 largest colonies in Florida Bay of spoonbills. And this is what's happened. 2005 at Sandy Key, there were 120 nests. 2010, 38. 2015, 9. Turnkey, where there was once 600 nesting pairs. 2005, 106 nests. 2010, I went into that colony and I about cried. There were no spoonbills there. And it's been that way ever since. We don't have any spoonbills nesting there. So where did they go? Well, that little green pin right there is a place called Madeira Hammock up on the mainland. And they started nesting there, and we estimate about 2005, but it wasn't really reported in big numbers till about 2009. It's really hard to get there. And so we tried to get out there in 2010, couldn't do it. Finally got in 2011, there were 164 nesting pairs. In 2015, 140. Also, Perota's Pond, another colony. 2005, there may have been a couple of nests, we don't know, but nothing to speak of. 2010, 25 nests. 2015, 128 nests. These birds are moving upland to where it's a little drier. So we look at the regional colonies. Tampa is all in Florida Bay up until 1973. 1973, colonies pop up in Tampa, start to really grow in 75. Merritt Island National Wildlife Refuge on the east coast of Florida, Cape Canaveral. Colonies popped up there in 1987. So at this point, the only places spoonbills were nesting in the state of Florida were in these estuaries. This is a map of those 3 areas. Now, this is also the historic range of spoonbill nesting in Florida. Is if you go back to pre-plume hunting era, this is where the nesting stopped. You would find spoonbills in North Florida, but they wouldn't be nesting there. This is the furthest extent they would nest north. So we take a look at the Tampa Bay total numbers, and all of a sudden in 2005, those numbers start to go down too. So this is a general pattern. So just looking at the colonies through time, decadal scale at a time. So 1936 to 45, all of the colonies were in Florida Bay. 46 to 55, Florida Bay. 56 to 65, Florida Bay. Now, 1973, a couple of birds nest in Tampa Bay. Then we see Merritt Island join the party. Okay, and this is where we see the first colony pop up in the middle of the Everglades. And that was, I think, in 1993. Now watch what happens. 1996, all of these new colonies are showing up inland. Even the ones associated with Florida Bay are inland. Even the ones at Tampa Bay are a little bit inland. This is to 2016. So these birds are smarter than we are. They're leaving the coast. But they're not just doing that. They're well north of that line from Tampa Bay to Merritt Island. They're nesting in Georgia, and we got reports that they're nesting in South Carolina. This is way beyond the normal range of spoonbills. So the Survival by Degrees report, this is what said the range was in 2000. And indeed, it was. This is what it predicted for in 2020 into Georgia and South Carolina. That happened in 2005. So sea level rise and climate change are really affecting this bird. And then there's this. Spoonbills in Minnesota, outside of St. Paul. That's not supposed to happen. So with that, I'll close and I'll just say that, yes, the sea level rise has changed the way things work in Florida Bay and in Florida. But it's more imperative in the face of sea level rise to follow through on Everglades restoration. These birds can fly, they can adapt, but things like crocodiles don't fly. And so you have to push that salt water out so it isn't such an abrupt change. So that isn't just going from freshwater to marine all of a sudden. And then allow it to gradually move so that the organisms there and the plants there can move with it. And it's also imperative that we continue to try and understand this. We restarted our satellite tagging program because we want to see where these birds are now feeding now that they can't feed where they were. We know they're still nesting in Florida Bay in smaller numbers, but where are they going? How are they surviving? And these are questions that will help us evaluate Everglades restoration. We can't expect the Everglades to look just like it did in the face of sea level rise. So we got to change our expectations a bit. What do we expect? And these kinds of studies will help us determine and set benchmarks for Everglades restoration and protection of these species. And with that, I'll be happy to end and answer any questions you have. Thank you very much.
Great, thank you so much. So I'm just going to start with the general questions, and then we have about 5 or 6 minutes for questions. And if we can some of the more specific ones, we will.
Oh, my apology for going over time, Erica.
No worries. It was such good information. I'm glad you did. I am putting in the chat our general email address in case you have additional questions. For those on Facebook Live, it is flconservation@audubon.org. Feel free to let us know if you have additional questions. But first, Jerry, why are spoonbills pink?
I don't know. There are 6 species of spoonbills in the world. I've seen 4 of them, and the only one found in the New World is this one, and it's the only one that's pink. All the other ones are white. I don't know why it's an evolutionary advantage. I really don't. We do know, you know, what the dyes are. You know what the colors are. We know the chemicals, but why these should evolve to be pink, I can't answer.
Well, how old are spoonbills when they can first reproduce?
About 3.5 years old, but we found with our tagging study that's variable. We've seen subadults. We have pictures of one spoonbill that was 20 months old, mating, and she produced young successfully. But it appears that most of the time it's about 3 years. It can be as late as 5. So just like in humans, it's highly variable, but somewhere in that 3-year range.
Burmese pythons are a very terrible invasive species in South Florida. Are you worried that they're going to affect nesting?
They already are. The wading birds here have no experience with them. As a matter of fact, and this is pure theory on my part, but the reason those birds weren't nesting in those wetlands where they are now is because they couldn't, the raccoons were just a terrible problem. So they would nest on these islands. Well, the pythons ate all the raccoons in Everglades National Park. And so that opens up new nesting areas. That's good. And some people, I've had the question, is that a silver lining? And it's no, because what it does is makes them python food. All of the wading birds, have been 27 species of birds, I think that's right, found in python stomach, including all the wading birds. So no, it's not a happy thing. And there's not much we can do about it, to be honest with you, until we figure out some kind of zoological treatment, a pest, a disease, something, some way to treat it. They're here.
How long do spoonbills live on average?
We guess about 25 years, and that is based on two things. Well, the one is we got that one bird that we know lived to 18 years. We've had a recent resighting of a bird in Tampa by Mark Rachal, our biologist in Tampa, Florida Coastal Island Sanctuaries, that was close to 16. And just by using the age of about 3 years of reproduction, there's a calculation that kind of predicts how long they would live, and that predicted like about 30 years. So I'm figuring probably 25 to 30, more likely in the wild 25. But we're still collecting data. So we know they live to 18, but our bands are, we started that program 16 years ago and we got a 16-year-old bird. So we'll see.
So are the colonies in northern Florida being monitored, and is the spoonbill's nesting success the same in Florida Bay as in the northern colonies that they've gone to?
I have a program going on in cooperation with the St. Augustine Alligator Farm. They have, that's a zoological park in St. Augustine, but they have a natural boardwalk that has attracted, because they have hundreds of alligators in their display, and nice cypress trees all around, a boardwalk that goes over. Well, as it turns out, wading birds love to nest over alligators. The alligators get a benefit because some chicks fall out and they get food. The birds get a benefit because the alligators will eat every daggone raccoon that comes along. And so they're attracted there. And so in about 2010, I believe, Spoonbill started nesting there. And so we've been monitoring them. We've been tagging them. And we started putting out some transmitters this past year. So we're getting that information. The ones in St. Augustine are doing tremendously well. We've already had a bird that I banded in Tampa Bay, nested there in 2010. Its offspring came back and nested there, I believe, 4 years ago. And so now we're working on, a third generation or fourth generation, and we're hoping those come back. And this gives us a whole lot of new information that we wouldn't have without kind of a pet colony, so to speak. You can walk right up to these nests and look in. The birds don't mind. So yeah, the answer to that question is they're doing very well.
So when is their nesting season? And just as a follow-up, can you talk a little bit about the 2018-2019 season? Was it a banner year for spoonbills like it was for some of the other species?
Oh no, yes, it was. It wasn't quite to that level. The problem with spoonbills is that we can't see them from the air. So when we had all those birds out there where we had, several 100,000 nesting pairs, the highest we've had in the modern era by far, you can't monitor spoonbills that way. They nest down in the trees, so you cannot see them from planes, which is how we monitor all the other wading birds. But even so, what we saw was a lot of birds, and we're seeing a lot of offspring. Those you can spot from the air. And so we don't have really good numbers. We just know that they did very well. In Florida Bay, they did not do so well, and that's that sea level rise story. So system-wide, they did well. I'm sorry, what was the first part of the question?
Just when is nesting season?
Oh, nesting season changes. In Florida Bay, it's typically from November to March. That has shifted with sea level rise. It's now January to April or May, because the water levels don't recede. So the birds just keep waiting and waiting and waiting for the fish to concentrate. But then as you move further north, like in Tampa Bay, it's more along the lines of January, February to April and May. So it's not static throughout the range. You know, they exist all the way from Argentina and Brazil up into Texas, Mexico, and then Florida and the Bahamas. So they have quite a broad range.
Are there any negative consequences for the spoonbills nesting farther north for them or other species?
Not that I know of. I just think that they're going to adapt. You know, I think this is what we're going to see, and this is the other report that National Audubon put out, is what will we see in national parks? And that predicted, for example, we'd see neotropic cormorants in Florida Bay. And one of my bird biologists last 3 years ago, found 3 pairs of neotropical cormorants nesting in Florida Bay for the first time. And so we're just going to see changes in where these birds are and how they do. You know, they're going to move from the coast, they're going to move inland, and more than likely, you know, most places are going to see a lot more. We're going to see a lot less.
Do the Florida and Texas colonies intermingle at all?
They have to. There are genetic studies done that show that there is significant genetic communication between all the different, somebody looked at Brazilian, Mexican, Texan, Florida, and the mitochondrial DNA, I believe, showed that, yeah, there is communication. But it also identified various parts of the gene that showed that they were indeed kind of, the birds go back to their colonies. Is different. You look at different components of the gene sequence, and you can see that these birds that are at this colony in Mexico are related. But there's enough spreading around. We estimate, based on our banding study, I didn't include this, that about 85% of the birds are going to go back to where they were hatched, and about 5 to 10% are what we call dispersers. For example, we had a bird that we put a satellite transmitter on that woke up one morning on Biscayne Bay in South Florida and decided, I'm moving to Andros Island in the Bahamas today. And it went there and it never came back. You know, so there are birds that go other places. We had that bird that I just said, hatched in Tampa, was one of the founding birds in St. Augustine. And so there are like 5 or 10% that move around quite a bit.
This is the last question, unfortunately. And like I said, though, check out our website for more information. Email us. We'd be happy to touch base. But Jerry, what can, you know, the average citizen do, or what can an interested person do to help the spoonbills?
Vote. That, in my opinion, protection of all these birds is the most power you can possibly have is who you vote for. Now, specifically, I have a lot of people who want to specifically come and do something for the birds, and that's a little bit harder in that, you know, it's very disruptive for the birds, especially during nesting season, to have a lot of people around. And so maintain your distance, respect the birds. If you go to a colony of nesting birds, you know, you so want to get close to take pictures. Maintain a very safe distance, because the predators, crows will come in and eat those eggs and those chicks like right now. And so be respectful. And support programs that, like Audubon, that you know, our goal is to evoke policy to protect these animals. So by helping Audubon, you're helping the birds.
Great, thank you so much. And for those tuning in on Zoom, I just also put a link. If you see a spoonbill that's banded, feel free to report it here.
Please.
Put that on the Facebook Live. And I just want to give a huge thank you to Jerry. This was so amazing. It was such great information. Like I said, we're going to record this, so if you want to go back and check something, you'll be able to do so. We'll send it to everyone who signed up. This video also lives on Facebook. And thank you all for tuning in. We appreciate your time.
Thank you very much.
Thanks, everybody.
Bye.